one of the more profound and interesting parts of the body and functions and how it works is the nervous system and cells. So, in this lecture, I'm going to talk a little bit about um, the biological basis of behavior, but I'm going to specifically focus on how neurons function and how it is they do the things that they do. Um, so we generally divide the cells in the nervous system up into two categories: neurons and glia, or neuroglio. Um, Neurons are the ones that conduct, transmit signals, and these electrochemical transmission of, um, your book says, and a lot of people talk about information being transmitted and so on. And I don't really like that word because it sounds like there's a message in there, but I think at one level that's really not what it is. The other types of cells in the nervous system are glia, and they are the glue, or the provide sort of the differentiation of things. Glia provide nutrients to neurons. They help protect and insulate uh, neurons, and they clear waste and so on. But relatively recent finding is we thought glia were sort of the dumb guys in the nervous system, and they didn't do much except sort of support. But it turns out that they may be involved in electrochemical transmission as well as in the signal processing in the brain. Neurons have some basic um, structures. This is just a, a cartoon from the internet that I pulled off. And basically, neurons have a cell body, or what we call the soma, and in it is a nucleus and a series of branches that we call dendrites, and then one long arm that's called an axon. And it's the message, the signal transmission goes down that axon to another, or in sometimes many other neurons. And so we call that place where one neuron sort of makes a connection with another neuron a synapse. So we call this so if the synapse is here, then the cell that's after the synapse is called the postsynaptic cell. The cell that's prior to the synapse is called the presynaptic cell. Now, there's some really neat things about axons. For example, axons are covered with the myelin sheath. This myelin sheath comes from Schwann cells or from astrocytes, and they have two functions. This is a fatty substance that protects the axon, and it also speeds in conduction. When you read that statement, you probably read it in your textbook, or you could read it online, that myelin helps with conduction. And it turns out that the fat substance that these Schwann cells, this myelin is made of, transmits an electrical impulse faster than the axon itself. So if we have an unmyelinated axon, down the axon at a particular rate, but this fat stuff allows it to travel faster. These axons, by the way, can be very long. We have axons in our body that are several feet long, believe it or not. A single cell can be several feet long. In fact, we have axons that go from the base of our spine to the tips of our big toe. A single cell, a single axon. These axons are very thin. What you see here represented in this cartoon is this. It looks like these are sort of fat sort of things, or big sort of things. They're really not. They're very, very small. They're 1/100th the size of a human hair or something like that. They're very, very tiny, very, very small um, processes. This is an actual picture of a rat brain, of a slice from a rat brain, and I wanted to show you sort of what it looked like in sort of real life, if you will. These black spots are actually the soma of neurons, and what you see here, these hair-like substances coming out, they're actually not axons, they're actually dendrites, and you can see that they expand all over the place. They sort of form these tree-like structures, and as you can probably interpret or see, they're making connections with each other. They're touching each other in this case. Um, these various features of this slice. This is about 60 times uh, microscope resolution. It's not great. It's kind of hard to sometimes visualize these, even with a great microscope. Um, but you can sort of get a sense of what's going on. These voids up here, these are probably blood cells, I'm sorry, veins or arteries that we're traveling through. You can see that there are other cells down here, other neurons that were also present, but since I've focused in on this particular level, what you see are neurons right near the surface of the tissue, and then these other dark spots are sort of a little bit deeper in the tissue. This was a 40 micron slice of rat brain tissue. To give you an idea, though, you can see all the processes. These are all dendrites from neurons, and doing important things. This is a more accurate and representative um, sort of cartoon of a neuron. Here is a neuron here. Here's the cell body. Here are dendrites. I'm not sure that the axon is represented here, but you can see all these dendrites have these little buds on them, and those are connections with other neurons. These are axons coming in and connecting with this individual neuron. So you can see the hundreds, and it turns out that many neurons in our brain, medium spiny neurons, for example, we call them, that have this like triangular shape, we call them pyramidal neurons, can have upwards of 30,000 connections with other neurons. So where one neuron connects another, or sort of touches another, actually don't touch. But they terminate. They get close to each other, and we call that a synapse when one gets close to another. And synapses, this is a particular cartoon that was talking about glutamate and dopamine and some other things. That's not really that important. But what you can see is that sometimes uh, neurons will synapse at these little buds. Sometimes they'll synapse on the axon, and sometimes they synapse on the soma. You can see that we get this interconnectedness between neurons at various locations and various places. This is another sort of representation or really simplified cartoon of what's going on in a synapse, and this is the presynaptic axon. So this is the message or the signals is coming down this way toward this one. This is the postsynaptic place or a dendrite where we're getting the message received. In the terminal buttons of an axon are located, we are contained in there are vesicles of chemicals we call neurotransmitters. And when this axon has an action potential, these vesicles, which are docked on the end of this terminal button, open up and sort of spill their guts into a space. There's a space in between these two neurons called the synaptic cleft, or the space between these two neurons is called this, where the synapse is occurring. This is called the synaptic cleft. The chemical travels across the synaptic cleft and binds to very specific receptors in a lock and key like format. This is a lock, and it is only opened, so to speak, by a particular neurotransmitter in a lock and key-like format. Now, synaptic vesicle activity is very interesting, and what we see is in this cartoon is a representation of that. Vesicles are manufactured and the neurotransmitter stored in the neurons, and they travel to the synapses. They're actually transported to the synapses, and they go through a process of docking and priming and calcium sensing and then opening. So the dynamics of this process is very interesting, and then they're translocated and refilled, or sometimes they're broken down. But this process is very interesting. It's very dynamic, and it's part of parcel to neuronal signal transmission. Now, on the other side of a synapse are receptors. So these, if we go back, we've talked about these things. So let's talk about these things right here, these receptors. This is where the locks are, and the keys are going to open them up. There are two major types of receptors that we talk about in the brain: ionotropic, or ligand-gated receptors, and metabotropic, or sometimes they're referred to as secondary messenger receptors. Now, the difference between these two is that they span the membrane. So in this particular cartoon, here's the extracellular space, here's the intracellular space, here's the extracellular, intracellular. Cytoplasm is what's inside the cell. Cytoplasm was what's inside the cell. So this is outside, this is inside. This is outside, this is inside. This particular, an ionotropic receptor, actually controls a channel for ions to pass through. So this is a glutamate receptor. So when glutamate comes in a lock and key format and binds here, then certain ions can travel into the cell, in this case calcium and sodium ions. You can see that there are other places where things bind to these particular glutamate receptors, but for example, PCP acts on them and so on. This is a dopamine receptor, and you can see that there's no channel really. In fact, it's not a channel. When dopamine binds to this receptor, it activates some other things inside of the cell. It doesn't open up the channel for things to come in, but uses another message. So the first message is the dopamine, and it sends a second message that does the work, and that's why they're called secondary messenger. So ligand-gated ion channels actually control an ion channel. When we have neurotransmitter binding to them, they open up, allowing bits of ions in, which changes the electrical dynamics of the cell. In G-coupled protein or metabotropic or other receptors, the neurotransmitters bind at the receptor, but their effects are through other messengers, which then control ion channels in a slightly different way. You can see through these. The reason these are called G protein coupled receptors is they have this protein base connected to them. When the receptors come in, it changes the dynamics of the receptors. The G protein releases a subunit, which then does its work. So let me try to describe how this works and the principles of neurotransmission, and why cells, for example, are negatively charged and in the interior and so on and so forth. So what I have represented here is a schematic with a cell membrane. This is the outside of the cell, and this is the inside of the cell. And let's say this is a blow up of a terminal button where there is some things are going to happen. We have things floating. We have ions floating around here with positive and negative charges. And in the cell membranes are what we know, we sort of refer to them as ion pumps. And what these particular units are doing is they are taking positive ions and pumping them out of the cell. They're pumping positive ions out of the cell. This results in more negatively charged ions in the inside of the cell. So therefore we have a negatively charged interior. We have a resting potential, we call it, of about minus 70 millivolts or so on. Now these right here are channels that would allow ions in if they were open, but the channels are closed. So the sodium ion pumps or the ion pumps are pumping out positive ions, resulting in a negative charge. Now what happens is if a neurotransmitter comes in and binds to a receptor, for example, in this case it might be a G protein coupled cell or receptor, it will affect the activity of a particular channel, or it might open up a channel, allowing positive ions to come in. As the interior of the cell reaches, let's say, negative 50 millivolts or so, okay, we reach a critical point when the actual membrane itself now changes. And what we wind up seeing is all the channels then almost instantaneously open as a result of this subtle change in the electrical dynamics of the cell, allowing a flood of positively charged ions in. And it almost instantaneous allows the cell to go from a negative state to a spike, a positive state. And that's what creates a firing of the cell, is that now we have a positively charged cell and it's transmitting this signal, so to speak. Now, very quickly, these positive and negative ions get sort of, they affect other processes. That's what I have represented here in blue and in yellow and orange. The positively charged ions do other work. They sort of change other processes, and then that leads to other things. But very quickly, what we wind up with is we're back to a negatively charged interior, a resting potential minus 70 millivolts, with a lot more positively charged ions on the outside than on the end. So hopefully that helped a little bit with how cells communicate and neural transmission of signals.